you very much, Kim. So my title of my talk is Asthma in the DNA Age. But first and importantly, I want to make the comment that science is about teamwork. And so a lot of the work I'm going to present is the result of, in some cases, collaborations with various national and international colleagues worldwide. But also importantly, it's the result of the hard-working efforts of a very important team of people consisting of students, postdocs, lecturers, etc. Quite a lot of the work I'll present has been published, but I'm also showing a few pieces of work that are unpublished, and therefore I've acknowledged the people involved in that on the slides. So I want to take you through first a little bit of history about DNA and the human genome. And it was in 1865 that Gregor Mendel, doing his experiments with peas, discovered the laws of genetics. However, it wasn't until 1944 that it was demonstrated that the DNA was the hereditary material. And then, in 1953, Watson and Crick and their colleagues in Cambridge, principally Rosalind Franklin and Maurice Wilkins, described the structure of DNA and the double helix that we're all familiar with today. However, it took another 13 years before they worked out how this DNA encoded the amino acids, the building blocks of proteins. Now, if we look at my timeline, my parents got married in 1954, and 7 years later had my older sister, and I appeared on the scene in 1967, and I apologise, I'm not going to show a baby photo. In 1972, recombinant DNA technology was developed, and I had my first day at school. Now, my school life was pretty typical, as I'm sure all of yours was, but during that time, in terms of DNA, a lot of things occurred. Sanger and Maxim Gilbert developed DNA sequencing methods, the methods that proved so useful in sequencing the human genome. The first human disease gene was mapped for Huntington's disease, and then in 1984 was the first public discussion of sequencing the human genome. In 1985, PCR was invented, and this became a very important tool for all molecular biologists, and especially geneticists. But in that same year, a young man called Bill Cookson returned from Australia, where he'd been doing his clinical training and also his MD by research, and joined Julian Hopkin at the O's chest unit. And Julian said to Bill, "Do a PhD with me. I think asthma is genetic. Let's prove it and let's try and find some genes." In the meantime, as Kim has highlighted, I was more interested in bacteria and the genetics behind those. And therefore I went to the University of Reading to do a BSc in pure microbiology. After graduating in 1988, I was not convinced that I wanted to remain at the laboratory bench, and therefore I took an administrative role at the Society for General Microbiology. I was responsible for looking into what microbiology graduates went on to do with their careers. It was a time in the UK where there was a considerable brain drain with people going from Britain, pure scientists, over to the States and America. I was also looking as to the existence of pure BSc degrees in microbiology, because the Southwood report had suggested they should be incorporated into more general biology degrees. I had the rather glamorous title of being the research assistant to the professional affairs officer, and this sometimes did cause confusions when people actually thought I arranged affairs between professors. Anyway, in 1989, I realised I really wanted to be back at the bench. And so I came to Oxford for a job, but not to the beautiful spires, but this rather more modern architectural building known as the John Radcliffe Hospital on the outskirts of Headington. And this is where Bill and Julian were based. I commenced work as a research assistant and then subsequently registered for a PhD, which I attained in 1994, with Bill attaining his a few months afterwards. And in that same year, in 1994, the Wellcome Trust Centre for Human Genetics was founded. And this moved into a purpose-built building in 1999. Now, in the meantime, Julian Hopkin has returned more to clinical work at the O's chest unit. But in 1999, he returned home to his home country of Wales, where he went on to establish the medical school at Swansea University. Now, as Kim highlighted, Bill and I continued on for a number of years up in Oxford until we realised the importance of relocating to Imperial College. And we came to the National Heart and Lung Institute in 2005. And then we were very delighted to receive last year the first Wellcome Trust Joint Senior Investigator Award, which gives us substantial funding to continue our research for the next 7 years. However, we were not quite so delighted to be the front cover of the Welcome newsletter. But now I'd like to talk about this period of time, a 13-year period from 1990 to 2003, when the Human Genome Project took place. After this was initially funded, in the first 7.5 years saw the establishment of the US Genome Centres and then also the Sanger Centre in 1993. So although many of you will be aware of the Sanger Centre, it's actually not been around that long. Now initially, less complex genomes were sequenced, for example bacterial genomes such as Haemophilus influenzae and the yeast genome. In the subsequent 7.5 years, saw establishment of 2 of the Chinese genome centres, and then finally in 1992, the sequence of the first human chromosome was completed for chromosome 22. Then in 2001, in the top science journals Science and Nature, the first draft version of the human genome sequence was published, with the mouse genome and the rat genome published in the subsequent year, and then in 2003 was the finished version. So now I want to try and give you a quick lesson in basic genetics and the central dogma. The human genome is composed of DNA, and this is organized into 23 pairs of chromosomes. 22 are the autosomes and 1 pair of sex chromosomes. And only 2% of the genome encodes for proteins, and this is organized into 23,000 genes. Now the DNA of these genes is transcribed into messenger RNA, and then this messenger RNA, through the triplet code, is translated into the amino acid blocks that then go on to form the proteins. But an important point about DNA is that during cell division and reproduction, the creation of life, DNA is constantly replicating. But during this process, errors can occur. The majority of time, the errors are corrected, but in some instances, they are not. And this is what we refer to as polymorphism. So this is sequence variation that occurs in DNA in every species, not only ourselves. The variation can exist in coding and non-coding DNA, and the variation can alter gene function. Now there's various types of polymorphism, and today I'm going to mention 2 types, so-called microsatellite markers, which are short tandem repeats of DNA. They're typically repeating units of 1 to 6 bases, and consequently there can be multiple forms of them, so 6 to 10 alleles, and therefore they are very informative. They occur about 1 every 6 to 30,000 base pairs in the genome. Now the other type of polymorphism is the SNP, which you're probably more familiar with, and this is a single nucleotide polymorphism. It's a single base substitution and consequently only has 2 forms. And with completion of the human genome sequence, we now know that SNP occurs every 100 to 300 base pairs. But why study genetics? Why am I here? Well, genetics not only allows us to understand disease, but also to classify disease more accurately. We can prevent disease in individuals at risk, as well as identifying new and novel targets that could be therapeutically addressed. And also, importantly, we can anticipate the response of patients to particular treatments. Now, asthma is a disease known as a complex disease. This means it's multifactorial. It's caused by a combination of environmental factors as well as changes and mutations in multiple genes. And consequently, it has no clear inheritance pattern. And complex diseases are typically common diseases in the population. So not only is asthma one, but diabetes, hypertension, and COPD. Now, asthma occurs in more than 1 child in every 10 in the developed world, and consequently it has a considerable financial cost and burden. There's an incomplete association with atopy, so-called allergy, that we measure through total IgE and specific IgE to common allergens. And atopic mechanisms have tended to dominate our understanding. Strong regional differences have been recognized. For example, if you grow up on a farm and are exposed to cows or pigs or drink unpasteurized milk, you have a lower risk of developing asthma. Similarly, ownership of a dog has been proved protective. And also, if you have an older brother, even though he may not be particularly desirable at times, he does provide protection against the development of asthma. So returning to polymorphism, the important thing about polymorphism is that it can act as a marker, allowing the identification and mapping of disease genes. And this is because of a phenomenon known as linkage disequilibrium. Now, if we look back at the chromosomes of our ancestors, if we think of just one chromosome pair, we can see here one is entirely blue and one is entirely white. But of course, over successive generations, due to recombination, mutations occurring, populations increasing and decreasing in size, and various selection pressures, when we look at present day, our chromosomes are made up of a mosaic of the blue and white blocks. So if, for example, A is a mutation in a disease gene, and we genotype this mutation, we will see linkage to asthma. However, invariably, we don't know about A. However, if there is a marker B that is on the same blue segment as A, if we genotype this marker, we will see linkage to the disease because of linkage disequilibrium. However, if we type marker C, this is no longer on the blue segment, it's now on the white segment, and is therefore not in linkage disequilibrium with A, and therefore we will not see linkage to the disease, and we will know that the disease gene is not close by. Now, before completion of the human genome sequence, there were two main methods for discovering disease genes. The first was known as the candidate gene approach, which was association-based and looked at known genes that were biologically plausible. These genes would be resequenced to identify polymorphisms in them, markers, and then these would be tested in cases, controls, and frequencies compared. Excuse me. The problem of this was there can be many candidates, and therefore to go through each candidate in detail and perform the genotyping was both costly as well as time-consuming. Nonetheless, there was considerable effort using the candidate gene approach. To date, there's 170 genes that are associated with asthma and its related traits such as atopy. And our group made considerable contribution to the candidate genes, identifying in a number of cases the genes and providing the first reports of association. So, for example, for NOD1, for the SPINK5 gene, TNF, and the HLA-DR locus, just to name a few. Now, the other method of finding disease genes pre-completion of the human genome was the so-called positional cloning. Now, this is linkage-based, requires no prior knowledge, and studies families. One looks to locate chromosomal regions that are inherited with disease, and then after that, through a process of fine mapping, resequencing, identifying genes in the region, testing SNPs in those genes for association, you'll ultimately, hopefully, find the gene itself. And previously unknown disease genes can be found in these regions. The attractiveness of it was more comprehensive, since you could examine the whole genome. Now, the first gene to be positional cloned was on chromosome 11, and this was the gene that we'd originally identified linkage to in 1989. But this gene proved quite tricky to pinpoint, and this was because there were a number of factors confounding the genetic linkage. Genetic heterogeneity, but more importantly, the maternal pattern of inheritance that we saw at this locus. So consequently, it took until 1993, 4 years, for us to identify the beta chain of the high-affinity receptor for IgE as the gene underlying the linkage that we've seen at this locus. Just prior to this publication, at the end of 1992, Weissenbach et al. Published a genome-wide map of 814 newly characterized microsatellite markers. So these are the highly informative markers I mentioned before. And therefore, we went on to do the first microsatellite genome screen for asthma and asthma-related traits. We identified 10 chromosomal regions of linkage, and others in the field reported a further 3. Now, for 8 of these linkages, due to considerable efforts by many people, the genes were finally identified. And our group identified DPP10 on chromosome 2 and PHF11 on chromosome 13. But what we noticed and thought was particularly striking, that was in 4 instances out of 8, the genes showed a bronchial epithelial pattern of expression. And also, for the 2 most highly replicated genes, candidate genes this is, filaggrin and SPINK5, they also showed an epithelial pattern expression. And for the first time, this really highlighted the importance of the epithelial barrier in asthma, as opposed to immune cells, which had been previously thought to be highly important. Now, once the human genome was completed in 2003, another initiative set off, and this was known as the International HapMap Project. And the aim of this project was to create a catalogue of common variants, SNPs, determine their frequencies and genotypes in various populations of different ethnic backgrounds, and also determine the linkage disequilibrium patterns between the SNPs. At the same time, there was a complete explosion in technology development, and we saw the microarrays being produced by two key companies, Affymetrix and Illumina. And these microarrays afforded us the opportunity to type 100 and 1,000, if not more, SNPs in a single DNA sample simultaneously and very rapidly. At the same time, array technology was also applied for looking at the contents of total RNA samples and measuring the mRNA transcripts present in the sample. Now, in the last 4 years, we've seen another explosion in technology development, and we now have the so-called next-generation sequencing platforms. And consequently, this has resulted in projects, I'm echoing, such as the 1,000 Genome Project, which in some way its name indicates what its purpose is, and in essence it's an American-funded project to sequence the human genome of 1,000 individuals. Now, with this fantastic new set of tools, we were able to conduct the first Genomewide Association study for asthma. We studied just under 1,000 cases of childhood severe asthma and just over 1,000 controls from 4 sets of samples of UK, German and Austrian background. And we typed them for just over 300,000 markers. Now, this is what is known as a Manhattan plot. Along the bottom we have the chromosomes, and along the side we have the log 10 of the p-value. So this is the p-value of association. So here a value of 11 equates to 10 to the minus 11. And each little circle is an independent SNP. We found multiple markers on chromosome 17q associated with childhood severe asthma at genome-wide levels of significance. We also saw a number of loci, but these didn't reach the genome-wide significance level that is standardly applied for studies of this type. However, this region on chromosome 17 contains more than one gene. So the question is, how do we identify which gene is the underlying disease gene? Now, we were interested at the time of using the array technology to do global genome-wide expression. And fortuitously, for 400 of the children that we'd included in the GWAS, we had also done global gene expression on RNA. Now, in doing global gene expression, this then allows you, if you've also got genotyping data, to test for association of SNPs to the level of expression of each of the 23,000 genes in the human genome. So we focused on chromosome 17. So this again is the region of linkage that showed association to asthma. And we asked the question, which genes in this region showed association to the same SNPs? And we found that transcripts of one gene, ORMDL3, also showed association to the same SNPs that showed association to asthma, but with p-values of 10 to the minus 23. And no other transcripts in the region showed association. Now, interestingly, ORMDL3 is again expressed in the epithelium, and it is an ER-based transmembrane protein. 2 years ago, Breslow et al., in their studies of yeast, identified that ORMDL3 is an important negative regulator of the sphingolipid pathway. It conforms a complex with serine palmitoyl transferase, which is the first rate-limiting enzyme step of the sphingolipid pathway. And members of this pathway, in particular ceramide, sphingosine 1-phosphate, and the lipids themselves, have been recognized to be important factors in immune signaling. Now, having completed this GWAS and also seen some hints that other loci existed, we realized we needed to increase the power of our study. And we were able to do this through the Gabriel consortium. So the Gabriel consortium was a Framework 6 integrated project that was funded by the European Union. And its main aims were to identify the genetic and environmental causes of asthma. And this whiteboard is actually the result of a meeting that occurred, I'm sorry to say, in Oxford before we moved to the NHLI, between myself, Bill, and 2 of our colleagues based in Munich, Erika von Mutius and Michael Gavesh. And this was the idea that we then generated into the Gabriel project. There was a work package specifically designated towards genetics, and we were fortuitous in securing further funding from the Wellcome Trust that allowed us to then conduct a second-generation GWAS for asthma. And this involved 10,000 asthmatics and just over 16,000 controls. Array technology to move further, so this time we could actually look at 600,000 SNPs and consequently generated 15 billion genotypes. Now, because multiple studies were involved, we were actually able to look at different syndromes of asthma, for example, childhood, adult, severe, and occupational. And in a number of these studies, total IgE measurements have been made in the subjects, and we use this as a surrogate for atopy. Now, the study involved over 150 investigators, and it was a fantastic collaboration. The investigators stretched from across Europe, included 3 groups from Russia, a group in Canada, and a group in Australia, and as well as people within the NHLI who collected severe asthmatic samples. And this is in essence what we found. So again, this is the Manhattan plot, but for those 10,000 cases and 16,000 controls, with the chromosome along the bottom position and the p-value up the side. And this marks the genome-wide level of significance. And as you can see, we saw 6 loci at genome-wide significance, the ORMDL3 locus on chromosome 17, but also a locus on chromosome 2, close to interleukin 18, the HLA region on chromosome 6, this is the MHC, an important inflammatory cytokine, IL-33 on chromosome 9, and also 2 further genes, SMAD3 on chromosome 15, and IL-2RB on chromosome 22. At a slightly lower level of threshold of significance, we also saw 2 loci which were independent of one another on chromosome 5, and IL-13 had been previously reported to show association with asthma, and also a further gene on chromosome 5 called RORA. We then asked the question, were the loci underlying childhood asthma different to those for adult? And we saw that the loci on chromosome 2, 9 and 17 were important for childhood onset, whilst chromosome 6 was important for adult onset. Now, for 7,600 cases and 7,000 controls, total IgE had been measured, and we therefore used this as a surrogate for atopy and looked for genetic association. We only saw 1 locus associated at genome-wide significance on chromosome 6, although at a lower level, lower threshold level, we did see a number of previously identified genes, the alpha chain of the high-affinity receptor, and also the interleukin-4 receptor, interleukin-4 being an important inflammatory cytokine. But importantly, none of these loci were associated with asthma, and similarly, none of the asthma loci showed association with total IgE. So what can we conclude at this stage? Well, in essence, genetics does work, but also asthma is genetically heterogeneous. IgE elevation is secondary to asthma pathogenesis, and interestingly again, for a number of the genes identified in the GWAS, their products are again involved at the epithelium, similar to what we saw with the positional clone genes. And this again highlights the epithelial barrier as an important and effective focus for therapy. Interestingly though, that genome-wide association studies to date for asthma have only explained 4% of the heritability. And this therefore demonstrates the presence of other effects affecting familial clustering. And these could be multiple small associations, dominance and overdominance, epistasis, this is known as gene-by-gene interactions, non-SNP polymorphism, and epigenetic effects. So we first try to address the issue of multiple small associations, and as you can anticipate, this means our previous GWAS of 10,000 is not powerful enough. And therefore, in September 2010, here in Imperial, members of the Gabriel consortium met with members of the EVE consortium. EVE was an American-funded study, again conducting a GWAS for asthma, and their asthma identified a number of genes in common with us. And so we decided to pool our resources in order to conduct a meta-analysis. Now, Bill decided that the consortium should be called MOMA, which stood for the mother of all meta-analysis. It has to be said some of our American colleagues were not so keen on this. I think the American mother, mama, is they're quite protective of. And so by the end of the meeting, much to Bill's disappointment, we had become the Transnational Asthma Genetics Consortium. At the end of last year, the TAGC actually had a publication. but it wasn't us. It was the Tobacco and Genetics Consortium, so whether we're going to remain the TAGC or be renamed again, we have to wait and see. Now, the Gabriel and Eve consortiums didn't want to be exclusive. We wanted to be as inclusive as possible, and consequently, there are now in total 18 consortia involved in this meta-analysis. This involves 67 studies in total, and myself with Professor Kathleen Barnes, who was one of the PIs of the Eve project, tried to coordinate this initiative. And together, the consortium has over 100,000 individuals with genome-wide genotyping data, of which 30,000 are cases of asthma. Excuse me. Now, the meta-analysis is being conducted by two fantastic statisticians, Professor Florence Demonay, who was one of the key statisticians for the Gabriel GWAS, and Dr. Dan Nicolai, who was the principal statistician for Eve. And the results are due, literally, in the next couple of weeks. Now, thinking positively that we are going to find further disease genes, the question then arises about how do we dissect out their function. And in our group, we do this by applying an omics approach. So if one considers this simple experiment that we routinely have performed before, where you take an inflammatory stimulus such as interleukin-1b and stimulate an epithelial cell, we typically will measure one or two cytokines as outputs, for example, interleukin-6 and interleukin-8. But if you think about it, this is highly simplistic, because in fact, when you apply stimulus to an epithelial cell, a massive amount of genes, pathways, and networks are switched on. And therefore, it's important to measure all of these. So omics and a systems biology approach is important because gene function is mediated by interacting networks of genes, and complex functions are mediated by co-regulated genes acting as dynamic modules. And we're very fortunate because now we have the omic platforms that allow us to systematically derive the data to identify the networks and the modules. So, for example, if we take the airway epithelial cell again, A549, and apply the inflammatory stimulus interleukin-1, what we do is we conduct a time series experiment. And so, yes, we can measure the cytokines IL-6 and IL-8 during the time course, but at the same time, in triplicate, we extract RNA and then perform global gene expression. We can then identify the genes that are up and down regulated, and then after this, we can cluster the genes based on their temporal patterns of expression. In other words, the way they change over the time course. So if we consider this experiment and look at the clusters we get, we found we got 6 clusters. So these little boxes along here, we have the time, and up here we have the change in expression. This is a log2 change. And what's particularly interesting is this cluster here, because we can see within the first 2 hours of the inflammatory stimulus, there is a 60-fold increase in gene expression. And when we look at the genes in this cluster, we see the familiar ones, IL-6, IL-8, a number of chemokines, transcription factors, but also there are 30 genes of unknown function. And these are important potential therapeutic targets. So we started applying this approach to the ORMDL3 gene. So this now is recognized to be the major genetic effect on severe childhood asthma. We've been making an ORMDL3 knockout mouse, which is another way of looking at gene function, but we've also been conducting the airway inflammatory cell experiment with the interleukin-1 stimulus. So again, here I'm just showing the levels of IL-8 on this blue line. It's similar to what I've shown before. But also, we've been knocking down the gene using siRNA. And I think you can see straight away this red line, which is the knockdown, results in a 70% reduction in cytokine levels. We've also, however, harvested RNA at the different time points in triplicate and run global gene expression arrays. And this is beginning to identify key networks and pathways that knockdown of ORMDL3 is perturbing. So I want to return to unattributed heritability again. So I've explained we're looking at multiple small associations. In the GWAS, the Gabriel GWAS, we were able actually to test for dominance and found no evidence for it. We could also look for gene-by-gene interactions for the loci we'd identified, and again, only saw incredibly weak evidence. And our attention is now turned to epigenetic effects, and in particular, CpG methylation. So DNA methylation is the most basic form of epigenetic modification. About 60% of all human gene promoters have sequences that are very CG-rich, so-called CPG islands. And the majority are unmethylated, and this correlates with an active status of the gene. You get transcription and expression of the gene occurring. However, if the CPG island is methylated, gene expression is repressed. So we've started doing genome-wide methylation studies. We've looked at 100 of our MRCA families, so these were families used in the Genomewide Association study, so we not only have SNP data for them, but also global gene expression. And we've treated the DNA by a process known as bisulfite treatment that then allows us to distinguish between methylated DNA and unmethylated using arrays such as the 27K array from Illumina. And methylation is measured on a scale of 0 unmethylated to 1 completely methylated. Now the data we've generated is highly complex, and so I'm going to just show a few of the key findings. The first finding was, if we look genome-wide, methylation is distributed in a bimodal manner. So again, this is like the Manhattan plots we've been looking at before with the chromosomes along the bottom, but the y-axis here is the degree of methylation, from 0 unmethylated to 1 methylated. And I think you can see that loci are either predominantly methylated or unmethylated, with a few in between. Now, because we had both SNP data and global gene expression data, and also a lot of phenotype information on these families, we were able to ask the question, what causes the variation in methylation? We found age accounts for 18% of variation in methylation, so-called NEQTLs. Sex accounts for 3%. Because we'd looked at families, we could actually look at shared family environment, and this accounts for 20% of the variation in methylation. Looking at the SNP data, we were quite surprised to see that only 10% of the methylation loci are affected by genetics, so SNPs affecting their status. More exciting, perhaps, was when we started looking at phenotypes. And for example, for total IgE, the top 4 methylation areas accounted for 20% of the IgE variation. And why this is so exciting, and I should comment, we've replicated this finding with samples from Julian Hopkins, based down in Swansea, is because methylation appears to have a much stronger influence on IgE than SNPs. For methylation, we're seeing it accounting for 20% of variation, as opposed to 1% for 1 to 2% for genetic polymorphism. Now, we're not unique in conducting studies of this nature, and I was fortunate to attend the American Thoracic Society in San Francisco in May, and Professor Donato Ficelli spoke from the Tucson, from the group based in Arizona, about the infant immune study that she coordinates and leads. And they started to look at global gene methylation. They'd taken 37 infants from whom they had cord blood DNA. All the infants were asthmatic, but approximately half of them had been born to an asthmatic mother, and the other half had been born to a non-asthmatic mum. And they performed whole genome methylation with a slightly different technology and asked the question, were there different methylation signals distinguishing between these two groups? And for 8 genes, they identified neonatal DMRs. Now, interestingly, one of the genes is DPP10 that we identified and positional cloned on chromosome 2, but what got me particularly excited was this gene, MS4A2, because in fact this is another name for the beta chain as a high affinity receptor. And 9 years previously, in our studies of the beta chain gene, we had looked bioinformatically, and I have to remind this was just as the human genome was being completed. We looked for CG-rich regions around and within the gene, and we identified 2 tracks, here and here, and this one was in the region that showed strong association with asthma, with atopy, but also with a maternal pattern of inheritance, suggesting that this tract is a substrate for epigenetic effects. Now finally, I'd like to talk a little bit about asthma and infection. Asthma has a complex relationship to infection, and infections cause exacerbations. And as I've highlighted earlier, it's been shown that a rich microbial environment in childhood is protective. However, I'm not a clinician, I'm a pure scientist. But my understanding is in clinical training, medics are taught that the lungs are sterile. Now, approximately 95% of bacteria are actually missed using standard routine clinical culture. And so people have started to ask the question how we can address bacterial identification using molecular techniques. Now, bacteria are highly heterozygous, but they do have a number of genes in common, in particular this one, the 16S ribosomal RNA gene, that encodes a component of the 30S ribosomal subunit, which all bacteria possess. Now, this gene is interesting because it has a number of constant regions, so constant in terms of their sequence, which means one can place PCR primers, and it also has 9 hypervariable regions, and it's these regions that can help us to identify bacteria present in the sample. So we can take, for example, a sputum sample, we can extract all the DNA present, we can then carry out this universal PCR amplification that will amplify a section of the 16S gene, including 2 or 3 of the variable segments for all bacteria present. We can then sequence these. Originally it was using cloned approach, now it's next generation. And then sequences we generate, we can interrogate against databases that contain 16S RNA bacterial sequences. And in that way we can identify not only the bacteria present, but also the communities. So we did this, we conducted a study where we obtained bronchial brushings from adults having COPD or asthma and healthy adults, and we also looked at sputum from children with severe asthma as well as healthy controls. Now the results we saw were the same in both sets of samples, so I'm just going to focus on the adult bronchial brushings, and this is in essence a summary of the findings. So we've got 3 rings here. The outer ring are the COPD samples, the inner, the middle, the asthma, and then the inner, the controls. And each of these coloured segments represent a different phyla of bacteria. And I think what is immediately noticeable, hopefully to all of you, are these orange and red sections are considerably smaller in the controls compared to those individuals with disease. And the phyla that are contained in these sections include pathogenic bacteria such as Haemophilus influenzae and Neisseria. In contrast, these purpley pink segments are substantially enlarged in the normal individuals compared to those in the COPD and the asthmatics. And these phyla contain commensal bacteria, such as Prevotella, which is an anaerobic bacteria. And anaerobe culture is not standardly used when in the clinical setting, which probably explains why people believed that the lungs were sterile. Now, we published this in 2010, but in fact we had the results in 2008, and it proved incredibly difficult to publish because many of the reviews came back saying, the lungs are sterile, you've got this wrong. And this probably reflects the fact that when the Human Microbiome Project was initiated in 2008, the lungs were not included as an organ of interest. However, it has to be said this hasn't stopped the respiratory community pursuing very proactively their studies of the lung microbiome. But why is the microbiome so important? For every cell in the human body, there are 9 to 10 bacterial cells on you or within you. So 10% of you, in fact, is human, and about 90% of you is bacterial. Now, obviously, we've been focusing a lot on the 23,000 genes in the human genome. But for every bacteria, it has a genome. And so if we actually consider the human microbiome, we're dealing with over 1 million genes. And therefore, it's very important that we start to consider the interactions between these types of genomes and how they are impacting on health and disease. Consequently, there has been a plethora of studies and publications and reviews, most recently a couple of weeks ago, in the top 2 journals, Science and Nature, who published the human genome sequence in 2003, with questions such as taking stock of the human microbiome and disease, microbiology, learning about who we are, and establishing what our fellow travellers are doing. And just since 2008, we've noticed a massive increment in the number of lung microbiome projects we're conducting in our lab, many of these with our collaborators at the NHLI and also groups at St Mary's. So in conclusion, I started originally as a microbiologist in the year that PCR was invented. And my team often laugh at me when I say you don't know how lucky you are, but literally this was the type of PCR machine I dealt with when I was doing my undergraduate degree, where we physically had to move for the different temperature stages the tubes between each water bath round and round again. However, then we saw the advent of the PCR machine, where we could put little tubes and let the machine get on with it. And this is actually the prototype that is on show next door at the Science Museum in the Health Matters exhibition. With this new tool, we were able to identify the beta chain gene of the high affinity receptor, so the first positional clone gene. PCR machines developed even further, such that we could now amplify up simultaneously 96 samples and even 384 samples. Microsatellite markers were identified, and so came the era of the microsatellite genome screens. Then in 2003, the human genome sequence was published. The HapMap kicked off, and we entered the era of Genomewide Association studies. And now our interest has turned to next-generation sequencing, methylation, in particular the beta chain of the high affinity receptor, and the role that the microbiome and bacteria play in the healthy lung and the diseased lung. And so I feel I've actually come full circle, but with an exponential increase in the amount of work now involved. Now, obviously, I need to say some votes of thanks. I want to thank the two institutions I've worked for, and I hope I will continue to be able to work for, Imperial College, and all the administrators, the heads of departments who've given incredible support over the years, the various funding bodies, MRC, Asthma UK, the EU, the Freemasons Grand Charity, but most notably the Wellcome Trust, who really have underpinned a lot of the research I've presented today. The fantastic Gabriel consortium with all the collaborators, both national and international, the new consortium, whether we remain being called the THEC, who knows, and all the other collaborators over the past 23 years. There are a few collaborators I do want to highlight: Professor Mark Lathrop and Ivo Gut at the CNG, who've been amazing support over the past 23 years. A group of statisticians, including Gonzalo Abacasis, who was actually originally a PhD student of ours, Liming Yang, Florence Demonet, Martin Farrow and Dave Strachan. The two individuals I mentioned, Erica von Mutius and Michael Kabesh, so key for the Gabriel Consortium. John Harper, who is a consultant dermatologist at Great Ormond Street. I haven't even touched today on what we've been doing in terms of allergic dermatitis, but again, lots of exciting things happening. And of course, my colleagues and members of the NHLI, the Molecular Infectious Disease Group led by Simon Cole at St Mary's, and those at the Royal Brompton Hospital. But as I highlighted at the start, science really is about teamwork. And I have to thank the past and present members of the Molecular Genetics and Genomics Group section. The present members are highlighted in bold here. It's just been a fantastic team of people. It's been a real delight and pleasure to have the opportunity to interact with them, to hear their ideas, to hopefully teach them to some extent, although I have to say now there are pure genetics degrees. I sometimes find it a little unnerving. But also I hope I've inspired them about how fantastic it is to do research and be in this field of work. But finally, and not least, I have to thank my research partner of 23 years, Bill Cookson, who has been both inspiring and equally frustrating at times, but has been steadfastly a very true and loyal friend and colleague. And I look forward to the next 23 years working with him. Thank you.